Wow. Well, welcome everyone to the February 2025 episode of the ongoing bi-monthly FR Panorama webinar series. Wow, I can't believe it's 2025 already, Mario. It's amazing
I
how
can't believe
fast
it.
time goes, huh? So I'm Larry Liberti, and together with my colleague Mario Alanis, as always, will be your hosts for today's webinar as we look into this very interesting topic of the value of early clinical data in supporting regulatory decisions. Well, as usual, today we have a lot of ground to cover, so we'll keep our introductions short. FR Panorama is an educational program of the RUD Foundation, a 501c3 nonprofit organization, presented in collaboration with the USC DK Kim International Center for Regulatory Science, which together bring you the FR Path project. We're also most grateful for the ongoing support by MSD for the FR Panorama series, and also to our advisory board, who provide insights and guidance with regard to topics that we cover within this series. You know, every year, a growing number of new medicines are developed and authorized for the treatment of all kinds of diseases that often represent a high unmet medical need. Developers, clinicians, and regulators, they all work together to meet urgent patient needs. But they're often faced with the opportunity to consider submitting and reviewing dossiers with clinical trial information that assess the product with what some call small patient numbers. Indeed, some applications have been based on fewer than 50 patients studied. Yet, a valid benefit risk decision was able to be made for these products. So rather than small numbers, I prefer to use the term fit for purpose numbers. Fit for purpose. These are data sets of high quality with strong evidence of a clinical effect that can justify a positive benefit risk assessment and an authorization. While many of these accelerated authorizations that occur from these data may be provisional, they may be conditional or otherwise subject to some additional verifications. So if I'm an agency that would like to rely on or be informed by a reference agency's decision based on unique data sets or novel study designs, what do I need to understand to inform my decision? Well, because even if a product decision is based on phase 2 or early phase 3 data, there can definitely be strengths in the data that allow a valid regulatory decision. So, to understand the issues involved in understanding how we've grown to be able to interpret early clinical data, to be able to make a benefit risk decision, today we're looking at this from three unique points of view. Mario, what do you think about this?
Yes, thank you very much, Larry. It's as usual, it's always exciting to be in these webinars. You know, I've had the experience of working closely with regulators who recognize the importance of these new therapies for their patients, but still they have a sense of reluctance or difficulty interpreting these novel types of data sets. So today, we're deep diving into how quality regulatory decisions are being made in this new world of clinical studies and data. And we have three very distinguished speakers who will address this topic. We have Dr. Tony Succar of the University of California, who will introduce us to the topic by explaining how we can extrapolate early clinical data for regulatory decisions. Then we will have Maxime Sasserville, who will share with us how the Health Canada agency uses phase 2 and early phase 3 data to support conditional authorizations. And he will be followed by Professor Marc Blockman, who will share the experiences of how the South African regulatory agency, SAFRA, sees the benefits and challenges of early clinical data for regulatory decisions. And finally, we will be joined during the panel discussion by Annalie Chu of AstraZeneca, who will provide reflections on this important topic. So let's get started, and I'm happy to welcome Dr. Tony Succar, who leads the Master in Clinical Trial Management program at the University of Southern California Department of Regulatory and Quality Sciences. We appreciate his sharing his experiences in working with a variety of clinical trial data. Tony, over to you.
Thank you very much for that very warm introduction. It is such an honor to be here and to present at this wonderful educational forum. Thank you so much. I started my journey in clinical trials back in 2006 in Sydney, Australia. So it has almost been 20 years in this exciting world. And just like Dr. Liberti mentioned, I can't believe how fast time has grown. And then also how fast clinical trials have advanced and continue to advance, which is fantastic. So I completed my master's and my PhD at Sydney University. I then moved to the University of California to complete my postdoc fellowship at the Keck School of Medicine. And then following that, I moved to Massachusetts Eye and Ear, the ophthalmology department of Harvard Medical School. And there I was managing all different types of clinical trials for drugs, for biologics, including stem cells and gene therapies, combination products and medical devices, as well as global multi-center clinical trials. After that, I was honored to return back to USC to join the Department of Regulatory and Quality Sciences as faculty of where I am today.
So thanks, Tony.
Thank
Take
you.
it away.
Okay, thank you, Dr. Liberti. So, extrapolating early clinical trial data for regulatory decisions is a very valuable strategy that can potentially accelerate drug development and approval processes, bringing not only life-changing treatments, but also potentially life-saving treatments to patients more quickly, and particularly those who have serious conditions with highly unmet medical needs. So this involves leveraging existing data from early phase clinical trials to make predictions about the drug's effectiveness, the drug's safety in a broader or different populations. So there are several methods which can be used for extrapolation. The major ones include the cross-design synthesis, and this combines data from different study designs, such as randomized trials and observational studies. It could also be used to supplement phase 2 trial data with real-world evidence, providing a more comprehensive view of the drug effects. So we will go into real-world evidence and real-world data a bit later in the presentation in more detail, but RWD can facilitate cross-design synthesis when combined with trial data to extrapolate results to populations initially excluded from the trial, providing early evidence of effectiveness in these groups. Other methods include reweighing methods, also known as standardization. So they adjust the trial population to match characteristics of the target population. So for phase 2 trials, for example, this could help extrapolate results to a broader and more diverse patient population. Discrete event simulation. This is a flexible modeling approach which can extrapolate phase 2 data by simulating patient outcomes over time. So DES can incorporate various factors affecting drug efficacy and safety, making it useful for predicting long-term outcomes. Parametric survival analysis. uses statistical modeling to extrapolate survival data beyond the observed follow-up period. As you can see, this can be particularly useful for estimating long-term outcomes from limited phase 2 data. Mixture cure models. These models can capture complex hazard functions by predicting survival separately for cured and uncured patients. So although estimating the cure fraction accurately is a challenge with limited data, they may be valuable for phase 2 trials of potentially curative therapies. Modeling and simulation can be used to extrapolate phase 2 data to support various aspects of drug development, including those of dose selection, the trial design optimization, and prediction of outcomes in different populations. So when applying these methods to phase 2 trials, it's important to consider the limitations of small sample sizes and shorter follow-up periods typically associated with these studies. Extrapolation results requires careful interpretation and consideration of the data quality, the relevance, and the appropriate statistical analysis to ensure reliable and valid results to inform regulatory decision-making. Some recommendations for implementation include engage with regulatory authorities early and frequently. Align early stage decisions with the applicable regulatory authority requirements. Focus on meaningful clinical endpoints. And anticipate and plan for potential obstacles ahead of time. Some benefits. Extrapolation can demonstrate early evidence of effectiveness in populations not included in original trials. This can help fill evidence gaps in the early marketing period. It can accelerate the development and review processes by aligning trials with regulatory expectations, and it can support regulatory and health technology assessment decisions. Many countries have pathways that allow for approved basis on surrogate or early endpoints, which can be derived from phase 2 studies. Some expedited regulatory pathways have allowed for drug approvals based on phase 2 trial data alone, potentially bypassing traditional phase 3 trials in certain cases. For example, the FDA's accelerated approval pathway allows for approval based on surrogate endpoints or intermediate clinical endpoints, which can often be demonstrated in phase 2 trials. The EMA's conditional marketing authorization enables approval based on limited clinical data, which can include phase 2 results with the requirement for post-approval confirmatory evidence. In Australia, the Therapeutic Goods Administration, or the TGA, has implemented 2 main expedited pathways. The Priority Review Pathway, which aims to complete the evaluation of a full set of clinical trial data in 150 working days, or approximately 5 months, compared to the standard 255 working days of approximately 8 months. So this is different to the FDA's priority review, which completes their evaluation in approximately 6 months compared to the standard 10 months. The priority review pathway of Australia potentially brings medicines to market up to 2 years sooner than the standard process, and if successful, the drug receives full registration on the Australian Register of Therapeutic Goods. The second pathway is the provisional approval pathway, and this allows for earlier market access based on preliminary clinical data. It aims to address unmet clinical needs and can approve drugs based on phase 2 data only. So this expedited pathway allows for earlier market access based on preliminary clinical data, and approval is granted for 2 years initially, with the possibility of extensions up to a total of 6 years. This enables time-limited registration of promising drugs based on preliminary, usually phase 2 data. Drugs can be brought to market, potentially used sooner, under previous processes, and it accepts applications before phase 3 trials are completed. The TGA only grants provisional approval if potential benefit of early availability outweighs the risks of incomplete data, and as a condition of approval, sponsors must provide a risk management plan and submit comprehensive clinical safety and efficacy data within the provisional time period. If confirmatory data are not submitted or do not support full registration, the TGA may revoke or not extend the drug's provisional registration. The integration of real-world data and real-world evidence with clinical trial data could provide a more comprehensive picture of a drug's safety and efficacy profile, potentially supporting regulatory decisions based on early clinical data. Real-world evidence is clinical evidence derived from an analysis of real-world data in routine healthcare delivery outside of controlled clinical trials, including electronic health records, billing and medical claims, insurance data, disease registries, and laboratory data. RWE complements traditional clinical trials by providing insights into how medical products perform in diverse patient populations and in real-world settings. So this plays a crucial role in extrapolating evidence from phase 2 trials, helping to address limitations and enhance the applicability of trial results to broader populations by allowing researchers to include patient groups often from underrepresented groups. Phase 2 trials typically have shorter follow-up periods, so real-world data can be used to model long-term outcomes and safety profiles beyond the phase 2 trial duration of extending the follow-up period, informing trial design by helping researchers understand the target patient population better and enabling the design of more inclusive and representative phase 2 trials. As phase 2 trials often have smaller sample sizes, real-world data can provide additional context and support for the observed effects, enhancing the robustness of findings and supplementing the limited data. Real-world data supports discrete event simulation by developing and validating outcome prediction models, which can then be used in simulations to extrapolate trial results to different scenarios and populations. By providing real-world context, real-world data helps improve the generalizability of phase 2 trial findings to diverse healthcare settings, further enhancing its external validity. Real-world data can support early assessments of potential drug interactions, helping to shape the clinical pharmacology strategies and inform future study designs. I'm going to touch a little bit on phase 1 and PK/PD modeling and the advantages, because the PK and PD modeling help to optimize clinical trial designs, potentially eliminating the need for certain studies. For example, it allowed researchers to skip a phase 2 dose finding study for a 3-month extended release formulation of paliperidone palmitate. Researchers utilized insights from the phase 1 study of PP3M to develop a comprehensive population PK model. So through this model and simulations, they identified a dose that closely matched the PK profile of the 1 month formulation in their phase 3 study, completely bypassing a phase 2 requirement. There are, however, ethical considerations, and regulatory decisions based on phase 1 data must prioritize patient safety and ethical conduct, including risk-benefit ratio, supporting further development, evaluating the appropriateness of patient populations for future trials, and assessing the adequacy of the informed consent procedures. There are limitations with phase 1 trials, and the smaller sample size, which is usually between 10 to under 100 participants, may limit the generalizability of the results. The focus on generally healthy volunteers in many phase 1 trials may not fully represent the disease target population, as well as the short duration of the exposure in phase 1 trials may not reveal the long-term safety issues. So these all must be taken into consideration. And phase 2 trials, the decision to approve the drug based on these data only is based on a case-by-case basis, considering factors such as the strength of the efficacy signal, the safety profile, and the severity of the condition being treated. For the FDA, most drugs go through a traditional approval process, as you're aware, which typically includes phases 1, 2, and 3 sequentially, and normally 2 phase 3 trials are required before FDA approval is granted. However, the FDA can approve a drug based on phase 2 data alone in certain circumstances, particularly for drugs that treat the certain serious conditions and fill the unmet medical needs. They are becoming increasingly common, and this is typically done through the FDA's accelerated approval program, which allows for earlier approval of drugs based on surrogate endpoints. This depends on whether the phase 2 trial shows strong evidence of the drug's effectiveness, such as high response rates and significant improvements in surrogate endpoints. And for these diseases with no satisfactory existing treatments and unmet medical needs, may be more inclined to grant accelerated approval based on phase 2 data. Drugs targeting serious or life-threatening conditions are more likely to be considered for early approval. A valid biomarker test and appropriate patient selection where targeted therapy can provide strong evidence for its value, even in an expanded phase 1 trial. It's important to note that drugs approved based on phase 2 data through the accelerated program are still required to conduct post-approval studies to confirm the anticipated clinical benefit. If confirmatory trials fail to show clinical benefit, the FDA has procedures in place that could lead to removing the drug from the market. The implications, this allows for earlier patient access to potentially life-saving treatments, especially in oncology and rare diseases. Regulatory bodies must carefully evaluate what constitutes sufficiently robust data, and through adaptive trial design, some phase 2 trials, such as randomized phase 2 trials, which are larger than traditional single-arm phase 2 studies, can generate sufficiently robust clinical data that might be used for full drug approval, potentially bypassing phase 3 trials. And so the final takeaways: approval based on phase 2 data is more likely for drugs treating serious conditions with limited treatment options. Drugs approved based on phase 2 data typically require post-marketing studies to confirm the anticipated clinical benefit. And while early clinical data is valuable, ultimately the regulatory decisions and approvals depend on factors such as strength of evidence, unmet medical need, and the seriousness of the condition being treated. Thank you, everybody, for your attention.
Wow. Thanks so much, Tony, for really helping to set the stage.
Okay.
I had no idea about the number of sophisticated extrapolation methodologies that are available, because we often hear about, let's extrapolate these data, but actually seeing that there are so many formal ways of doing this was just eye-opening to me. And that especially being able to extrapolate where there weren't even patients in a particular category. So thanks for telling us about those approaches. Really
Thank you very much.
appreciate it. Yeah.
Thank you.
Well, as
Thank
I
you,
mentioned
Tony.
earlier, many drugs are being approved with some form of qualification associated with them. And importantly, conditional marketing authorizations, which we hear a lot about, these vary in name around the world. We just saw some that Tony was talking about. But they all rely on early clinical data and seek some kind of confirmation through growing clinical experiences, as Tony just mentioned. Now, among the agencies with much experience with conditional marketing authorizations is Health Canada. And today, we're really pleased to be joined by Maxime Sasserville, the Acting Associate Director of Pharmaceutical Drugs Directorate in Health Canada, who will give us some insights onto how his agency views these assessments. Welcome, Maxime.
Thanks, Larry. It's a pleasure to be here presenting today. I'll be I'm currently associate director with the Bureau of Metabolism, Oncology, and Reproductive Sciences, but my substantial position is in the oncology division as a manager. We have 4 oncology divisions, and I'm leading one of the 2, making the pre-market review of pharmaceutical drugs. And okay, here are the slides. Thank you. So that's my disclaimers. So today I'll be speaking a bit. It's going to be very complementary to Tony's presentation, I think, because we're going to be talking about the regulatory review very briefly at Health Canada, focusing on oncology products, and then I'll give you sort of an overview of our program of conditional of notice of compliance with conditions. So this is our organization, Health Canada. Within Health Canada, the Health Product and Food Branch is the organization responsible for reviewing new health products. If you look at the central column, Pharmaceutical Drug Directorate is where I sit with new pharmaceuticals, but you also have a directorate for biologics, radiopharmaceuticals, one for non-prescription drugs, and one for medical device, with the Marketed Health Product Directorate providing a horizontal safety monitoring for all health products in Canada. So we regulate about more than 13,000 drugs in Canada under our under Health Canada's purview. And basically, our regulatory framework comes from the Food and Drug Act and associated food and drug regulation. So under the Act, what you need to market a drug in Canada is you need to submit a submission that is getting reviewed, and you need to obtain a drug identification number and a notice of compliance to sell your drug. To obtain that, basically manufacturers have to show or to provide sufficient evidence of the product's quality, safety, and efficacy as required under the Act. So here my it's my oncology bias that shows is oncology special compared to all the other therapeutic area. I'm biased, but I'd probably say yes. The regulatory process are pretty much the same, but oncology uses some of those processes more often than other therapeutic areas, as you'll see in the coming slides. We have patients in oncology trials have a different risk tolerance, just basically due to the life-threatening nature of the disease and basically their desire to be cured or to prolong their survival in the palliative setting. So that's basically the area in which we have seen the most development of innovative regulatory approach. So I'll talk briefly about two of them, and I'll concentrate then on the notice of compliance with conditions. So the first one, expedited program, is the priority review. So the status is granted to new drug submissions and supplements for a new indication, and provides a certain review timeline. In Canada, the standard review timeline is 300 days. You can shorten it down to 180 days for priority reviews, and this is granted for following a formal request, for drugs that are intended to treat, prevent, and diagnose serious and life-threatening disease and conditions for which there is substantial evidence of clinical effectiveness. And please note the substantial evidence, because it's one of the key differences with the NOCC pathway. The substantial evidence means that there's less support coming from surrogate endpoint, surrogate markers, and more established clinical endpoints. On the flip side, you have the other expedited pathway, which we'll talk more about, the notice of compliance with conditions. So basically, the drug needs to treat disease and conditions similarly to the priority review, but the big difference is that it has to qualify under the NOCC policy, is that it has to provide promising evidence of clinical effectiveness, and that's where you see a lot more early data coming in, with more surrogate endpoints being the support. So basically, once we approve an NOCC, however, we do require confirmatory evidence, but we also require enhanced monitoring and reports on safety and efficacy of the promising new therapy through enhanced post-market surveillance activities. So basically, what I'll walk you through here is an analysis done by the group of Calvin Chan from Sunnybrook Research Institute in Toronto, Canada, to which colleagues from Health Canada have participated. And basically, it describes or reviews basically all of the NOCC approvals that have been made since the policy's creation in 1998. We have approved since then 149 new drugs, generic drugs, and new indications under the NOCC policy. And I think you can see here by the graph on the right that there is about a 1/3, 2/3 distribution of non-oncology indication and oncology indication. I think it's interesting to note that when it was created in '98, there was about a 50-50 proportion, or even less, maybe predominantly, for example, AIDS drugs that were being approved under NOCCs, and always a small numbers of oncology drugs. In recent years, if you look at the percentage now that has grown over the years, we now have overall about 70% of drugs that are approved under NOCCs that are in the oncology field. And if you take the last period that was reviewed by this review, 2018 to 2021, 95% of NOCC decisions were for cancer indications. So we're predominantly the frequent flyers for the policy. So when we talk about notice of compliance with conditions, what we're interested in is the time is how realistic and how successful they are at generating the confirmatory evidence. So what we see on the right hand side, I apologize for the small font size here, but on the y-axis you have the percentage of NOCCs that have fulfilled their confirmatory evidence commitment, and that's a sort of a Kaplan-Meier curve, or equivalent to a Kaplan-Meier estimate. And you have in blue the oncology drugs, and in red, the red curve, the non-oncology drugs, as a function of time. So what you can see is that actually the time to generate the confirmatory evidence is rather similar between non-oncology and oncology drugs. And you have a median time to meeting your conditions for oncology drugs of 53 months, or a little more than 4 years. You can see that after a year post NOCC, there's virtually no one meeting their conditions. Whereas when you hit the 5-year post NOC mark, you're getting to a majority of NOCCs being confirmed and issued a full NOC. The second part also about the conditional approvals is sometimes there is misalignments between the initial or differences between the initial the setting in which the initial approval was granted and how the confirmatory evidence are generated. In a majority of cases, you have an alignment between the indication, the initial trials, and the confirmatory trials patient population. So in 67% of NOCCs issued, but you also have another cohort of NOCCs, that represent 18 or 19% of them that are partially aligned. So meaning that there is differences between initial and confirmatory trials in previous therapies, for example, cancer types or biomarkers. And the last bit is that sometimes you just don't have an alignment with the initial and later confirmatory trials, and that's due mostly to the different treatment lines in which the therapies are being looked at. The other point I wanted to make is the endpoints and the study designs that we see at initial approval and confirmatory trials. If you look, again, that's a little small, and I apologize for that. If you look at in the graph on the left side, you see the initial trial designs. What we can see here is that the uncontrolled single-arm trials represent the majority of the trial designs. So about 73% of oncology drugs are approved based on either uncontrolled or phase 2 trials, and that's illustrated by the median sample size of 249 patients, which is relatively small. What we see also as a majority of endpoints being used in those uncontrolled trials is, in a majority of cases, overall and objective response rates and duration of response. And you see also when you have early phase 3 trials, randomized controlled trials readouts, sometimes you will see progression-free survival and disease-free survival, but rarely overall survival at that point. Whereas if you look at the right side, of the curve with the designs of the confirmatory trials, you see that phase 3 trials now represent the majority of the evidence being requested and being provided as conditions to meet for the conversion of the conditional approval to full approval, and that's in 84% of the cases. And you see that the median sample size goes up to 702, which is more more representative of this stage of development. But nevertheless, there was still just like 33% of confirmatory trials in the confirmatory stage that included OS as an endpoint, and 47 or 48% that included at least one trial reporting PFS as a primary endpoint. So overall, what we're interested in also in the NOCC is basically how successful it is, because when an agency relies on early data to approve a drug to allow it for sale on the market, is you want to manage the risk of approving a drug that turns out to not be superior to existing standard of care. And we ran the analysis with the 34 NOCCs issued between 2018 and 2021, which are for oncology drugs. And what was seen was that in close to 80 cases, the drug that we approved under NOCC turned out to be ended up being recommended by the national the NCCN, National Comprehensive Cancer Network recommendation for their respective indication, suggesting that the many of those therapies we approved on early data ended up being recommended therapies in clinical guidelines. And 47% of these, they ended up being preferred regimens, so the gold standard for treatment, and some others being either other recommended options or useful under certain circumstances. So and in 10 of them, they were just simply not categorized. Only 5 of the 37 NOCCs ended up not being listed in the NCCN guidelines. So to date, if we account for about the 100 therapies that were approved in oncology for under the NOCC policy, only 4 have been suspended or discontinued due to clinical trial results that indicated increased risk or lack of efficacy. In addition to that, 3 NOCCs ended up being withdrawn by the manufacturers before their conditions were fulfilled. So a few take-home message. Health Canada knows of compliance with conditions policy, despite relying on earlier clinical trial data, often also based on surrogate endpoints and fewer patients, provided expedited access to promising new therapies for serious conditions. The analysis also indicated that most new oncology therapies approved under the NOCC policy turned out to become standard of care treatments for their respective indication, in a vast majority of cases. Companies also planning, and I think I will sound very similar to Tony. Companies planning to use this pathway should consult early with regulators to discuss their potential confirmatory evidence generation program. I think that's very key to make sure that the regulators have the data they need and can verify the early signals of effectiveness that we see in early trials. And last, ensuring that confirmatory trials are underway at the time of NOCC issuance is very strongly recommended, as studies addressing the uncertainties associated with the NOCCs should be completed in a timely fashion. And with that, I leave with you with a few resources for if you want to have more information. And I thank you very much for your attention.
Thank you. Wow, very interesting. Thank you very much, Maxime, for explaining how, in reality, Health Canada uses early clinical data and the level of rigor you apply to your assessments. But now we're going to see, what if you are an agency and you would like to rely on a decision of a reference agency to inform your own reliance decision about a product that may have been approved with only phase 2 or early phase 3? Agencies are interested in understanding what are the risks that they are assuming by doing this. So they want to understand, how do you approach understanding the reference agency decision? How can they apply that decision to their own population? Well, to give us insights into this topic, we are honored to have today Professor Marc Blockman, who is representing SAFRA, the South African Regulatory Authority, and it's also director of the WHO Collaborating Center for Medicines Information of the University of Capetown, and he will bring his experience to our conversation today. The floor is yours, Professor.
Thank you very much, Larry and Mario, and thank you very much, everybody, for joining us. My name is Marc Blockman. I work in internal medicine at Groote Hospital in Capetown, South Africa, where the first heart transplant occurred. And I'm representing the South African Health Products Regulatory Authority, and because I chair the Pharmacovigilance Committee, to say that we're very new on the block in terms of these thought processes. So I'll give you some of our experiences and what we're thinking about at the moment. So thank you very much. So just to remind us that obviously that clinical trials are critical in terms of making decisions for medicine regulation, because they provide us with evidence of safety and efficacy. The problem is that while the gold standard is phase 3 clinical data for regulatory approval, there's been a move now, which is really important, to try and use early phase data to help make our regulatory decisions. And could we use some of that data and resources which are being done by other regulatory environments, especially to look at medicines for unmet needs, for example, orphan drugs, for example, and there was a lovely example for oncology. The issue for us is that we don't have at this moment in time in the South African health product regulatory environment a legal framework for handling such products and such decisions within our own environment. And so we are taking the teachings and learnings from other environments and looking to upskill ourselves in terms of these decisions and the way we review dossiers and the way we review new medicines. So just to remind us, as you suggested, is that the phase 2 provides the early evidence. Phase 3 gives us obviously expanded populations, and we've heard that a lot about the numbers and how much we need. But in fact, one of the introductions is around having good studies, well-targeted studies to give us the information. And as I suggested, is that SAHPRA doesn't have this formal framework at this moment in time for reviewing medicines for unmet medical conditions. And so we are having to rely on our reliance programs with our partners. So this has been key in terms of our discussions and our decisions around the key benefits. And the important for us is around access to medicines earlier within the chain in terms of achieving potential clinical benefits, and in that way providing our population early on with medicines which may have taken a bit longer to come to market. And so we're hoping that accepting clinical data from these reliance decisions provides this to our population. So this is part of our thought processes around what we would be looking at in terms of, one, using reliance data, but also as we upskill ourselves in terms of making these decisions. And well, we've heard already that robust study designs are really important, using adaptive trials and real-world data. And there's been a fear in sub-Saharan Africa to use real-world data because we're not always sure on the unbiasedness of that. But I think that this is a critical aspect for us to learn about in terms of utilizing that in our regulatory environment. Importantly is to engage early with regulators, seeking guidance on acceptable endpoints so that there's a shared decision on what endpoints we believe are clinically relevant, which are looking at efficacy, and for me, importantly, is safety. And then importantly about biomarker integration into the studies, and making sure that we can predict outcomes of efficacy and safety using appropriate biomarkers. And that requires obviously strong clinical acumen to do that. And then critical, using big data, leveraging artificial intelligence and modeling to predict our long-term outcomes from some of these early data. And really in terms of POP PK, this is already occurring within the environment using sparse sampling to determine PK modeling. So some of the case studies that we've had, and we've basically relied on the COVID-19 vaccines being one of our test cases for using early data to make decisions and for emergency authorizations. We had a lovely conversation now on oncology drugs, biomarker driven in terms of accelerated approvals, and then rare diseases. And we don't have a policy within the country for rare diseases, despite the fact we have many of our populations who fall into this category. And we certainly need to rely on the robustness of review by other agencies so that we can make early decisions on risk-benefit. And so what our teachings have been is in the COVID-19 vaccine discussions, which were in our traumatic recent past, in terms of understanding the unmet medical need and having to be flexible and a leap of faith in terms of the consideration of using very early data, which was not mature in terms of making decisions. And what was really important and celebrated is how there was so much sharing between the regulatory environments to provide these early considerations to be adopted or adapted within the environment. And for us, safety was very important in terms of prioritizing life-saving products in our environment. And we put in place strong pharmacovigilance, public and healthcare worker centered to try and make sure that we improved case findings for safety. Another thing that happened in sub-Saharan Africa is countries got together to develop a joint pharmacovigilance process of trying to have early signal detection within our own populations. And so we had flexibility in our within the crisis. We required to use real-world evidence and to have that shared real-world evidence. We became more flexible in terms of using early phase clinical trial data to help make our decisions. And we were able to provide, basically manage the approval process from a legislative point of view to find an earlier temporary approval process to allow us to be able to use some of the medications, but certain vaccines in our environment. And so we created a fast-track system for reviewing the clinical trial applications, and that did a few issues in terms of making sure we were upbeat and skilled in terms of some of the medicines and vaccines that were being trialed, but also relying on market authorizations from other environments to make sure our population were benefiting in terms of medicines or vaccines. And we then developed guidelines on how to incorporate these real-world data into emergency use. And importantly, we collaborated. We worked closely with the WHO, EMA in this situation to expedite the approval process, learning from some of those environments in terms of making early decisions, temporary early decisions with a shared responsibility. And so we were able to provide conditional approval for using early phase clinical data to make our decisions, and hopefully that robust, mature data consolidated that information and that we were on the correct path. So some of the key challenges that we are thinking about and how to move forward is the limited sample size. Small patient groups may not represent the wider population. There's variability in data, and we've seen that inconsistent results can complicate the regulatory assessment. Regulatory uncertainty, as different agencies have different requirements for early data use, and we've got to make a decision of which agency we're going to use and rely on that decision to use their thought processes on early data. Importantly, to get our heads around the lack of long-term safety and efficacy is very important for a regulator. And then wearing my own hat, the need for a mature post-marketing surveillance process built into making these decisions, both active and passive surveillance, as well as risk management plans to make sure that medicines are used safely and that we pick up the signals. Some of the variability in early data outcomes is that differences in patient responses can make it quite difficult to determine efficacy, and one has to trust that what we're seeing in one patient population will translate into the patient population more globally. Small trials may not detect rare side effects, which we certainly found that occurred in the COVID-19 vaccines, and that we required robust risk management plans to pick them up and to act quickly and smartly in terms of making a better or quicker regulatory decision. And the regulators require consistency before advancing approval. So we're required to make sure that we have a systematic approach, that we're consistent to what data we rely on, and so that doesn't provide confusion into the market. And that also means that we are that our own regulatory thought processes hold us to be accountable as well. Some of the key SAHPRA challenges that we are battling with at the moment is that regulatory decisions on early clinical trial data may be statistically significant but not clinically significant, and we have to appreciate that and look to work around that. Animal data do not always correlate with human data. That's been shown many times. And then for our populations in sub-Saharan Africa, and especially in South Africa, is the burden of HIV and TB. Many of our population use antiretrovirals and anti-tuberculosis agents together and what are these drug interactions look, drug disease and medicine interactions that won't be studied early on when we are making these decisions. And it's again a leap of faith that certain populations are not excluded from management because they may have comorbidities that we haven't thought about. As I've said, we don't have a legislative framework for conditional registration, and so we are working on that to come up with a way forward to be able to do that, and so that it makes us more reliant on the reliance process for early regulatory decisions around registration. And in resource-constrained settings, the lack of solid pharmacovigilance systems may lead to inadequate regulatory oversight. And so there's certain information that you can certainly achieve from the global north, but there may be aspects that are prominent only in sub-Saharan African populations that we may miss with a resultant clinical mayhem. And so in conclusion, we note the importance of early clinical data in terms of regulatory decision-making and making medicines more accessible quicker to our population. We have certainly recognized that it enables faster drug development and improved patient access to innovative treatments. And I think we celebrate that, and our population will celebrate that as well. I think continued collaboration between industry and regulators is essential, and between regulators and regulators themselves is absolutely critical to make this work and for accountability and trust within the process. We need to develop an adaptive regulatory framework to allow for these accelerated review processes, while importantly maintaining the rigorous standards of safety and efficacy. And we are taking these teachings and learnings from more mature environments into our own environment at the moment. And again, this collaboration and reliance between regulatory agencies are critical in terms of enhancing the speed and safety of decision-making in not only public health emergencies, but in medicines for unmet need for our own population. And I thank you very much for your time.
Marc, I want to thank you. That was just a wonderful conversation about this very important topic about how to make decisions based on other reference agencies and understanding how they use data and how to interpret whether or not it's relevant to your population. We'll come back to that in the panel discussion, and also how you use the COVID-19 experience to learn and develop procedures. You're watching the February 2025 episode of FR Panorama, where today we're discussing the use and value of early clinical data in supporting regulatory decisions. Well, now we have some time to reflect on all of the topics that you've heard today, and we'd like to bring our speakers together again. And to get things started, I'd like to introduce our guest panelists, and including Dr. Anna Litziu. So Anna is the Regulatory Policy and Intelligence Director at AstraZeneca. And Anna, glad to see you. Happy you could join us. You know, Anna, what's captured your imagination from today's presentation? What are some of the key things you're thinking about from a regulatory policy standpoint?
Yeah, I think, thanks, Larry, and I'm glad to be back. And we managed to, go through the technological challenges. So I think one of the key things from the three speakers, and especially from Marc from Succar and Maxime from Health Canada, I think one of the key things is about collaboration. So I think from a policy perspective is, collaboration, because, working in these areas, we're talking about, novel mode of actions, novel modalities, for example. And I think from a policy perspective, one of the key things that Marc did touch is that when you are an agency where you are relying to other reference agencies, to other more mature agencies where they have these frameworks and they apply, the principles of conditional approval, they have seen some heterogeneity in the way that basically they practice. So I think from a policy perspective, through collaboration is, looking the spectrum of what we have been learning is how we can harmonize. So then we can help also, the agencies that they look up to, agencies like Health Canada, US FDA, or EMA to work in a more harmonized and collaborative way. Yeah, and I think the other key thing is about capability build, and I think it was echoed through through I think from Tony, Marc, and Maxime, early engagement, basically sharing your science, and I think that will be need more because we are in a very interesting crossroads with lots of new modalities of action, but also. Diseases, as we learn more and more biology, especially with the cell and gene therapy out there.
Yeah, those are really important considerations, aren't they? To be able to think about standardization at some level, so that there's more predictability in these processes, because they are important. And that's actually a question I have for our speakers: is that, with the changing mix of products that are being developed, we're developing advanced therapeutic medicinal products and cell and gene therapies. I have a feeling that we're going to see more and more use of these very specific kinds of pathways, these conditional pathways, accelerated pathways. In fact, we'll talk about it. We're going to be talking about these in more detail in about 2 months at our next webinar. But, tell me from your points of view, and maybe we'll start with Marc, and then we'll go to Tony and Maxime. How do you think the regulatory arena, the regulatory atmosphere is preparing itself for this growing potential need for these kinds of pathways? So, Marc?
Yeah, so thank you for that. I think critically is that one, we have to be flexible, and two is that we have to keep abreast of technology and what is occurring in terms of more directed therapies that for patient populations, and importantly is to look at the clinical trials that are emerging. So you can't do this in isolation. I think that people within the regulatory environment must maintain their own skills in terms of interpreting clinical data, must understand where the industry is moving in terms of, or where the world is moving in terms of personalized medicine, for example, and earlier therapy, understanding that the risks of those therapies may only translate many years down the line in particular patients, and you won't be able to wait to be able to be doing gene therapy, for example, which we're seeing quite a lot of studies at this moment in time. You can't wait for the safety data to be mature to make a decision. And so one needs to understand what preclinical data looks like, what the epidemiology of the disease is. And so we need to better upskill ourselves and be more flexible in terms of our regulatory processes, and then collaborate. I think we have to rely on each other's skill sets and technical abilities and review processes to be able to be able to make these decisions early on.
Interesting. Yeah, I see a theme coming out here, and with regard to collaboration and early conversations. And Tony, what's your opinion about the way the clinical trials will be used as we progress through these evolutions of new kinds of products?
Yeah, thank you very much, Dr. Libretti. So I think, as you already mentioned, the new treatments are complex, and the clinical trials complexity have to match, because there are certain treatments that certain designs of clinical trials are best suited for that particular treatment. For example, basket clinical trials, umbrella clinical trials, platform clinical trials, etc. They all have specific advantages that can be used for that specific treatment. And so regulatory authorities, as Dr. Blockman mentioned, need to be flexible, and also now they are becoming more flexible in accepting different design types and very complex and specific clinical trial designs to help show the evidence of those treatments. And I think that more research and will go into the designs, and those designs will continue to evolve as to match the evolving treatments.
Yeah, this is a very dynamic area,
Yes.
isn't it? In
Absolutely.
trying to develop that. You know, Maxime, you had shown how the use of the NOCC was increasing, and that it was specifically increasing with regard to oncology products. And if you look to the future, how is Health Canada kind of preparing itself for a growth perhaps in the use of NOCCs? And are there any recommendations you might be able to give to other agencies that are developing conditional authorization pathways that you think might be helpful to them?
I mean, as the field has evolved in recent years, and we've seen an increasing amount of use of very specific biomarkers, for example, like very narrow, rare patient population that are that bear certain mutation that for their tumors to have a certain characteristics. I think we've seen the number of patient, the size of the potential patient population shrink, and that has required us to sort of adapt and to look at more and more at those approach. There is area where resource-wise, a bit like Marc, we struggle, and a regulatory framework, it can only be bent so much. Like, they're not built to be very easily changed, and basically we have to be amenable and receptive to those new environments, those new kinds of designs. We have pathways, for example, that we call the advanced therapeutic pathways. It's basically a sandbox where we're trying to understand how to best regulate new products, very innovative, very cutting edge products. And we have to learn together with the evolving science. So we have those kinds of programs that we have so that we can receive from industry the kind of new the new edges of knowledge that they are developing on, and to advise other, I mean, for other regulators. I think we're all trying to just keep up with the evolving science. I think that's the critical challenge of an organization, is to onboard new employees, have new expertise, and yeah, I really like the collaboration edge. It's something that we work very extensively in. We have a couple of networks, for example, the Orbis project with the FDA, Brazil, MHRA, Swissmedic, TGA. Like, there's a number of those forums in which we can exchange information and share expertise, and just like help each other out. So I think that's definitely a key part of making sure we're keeping up with and staying up to speed with the evolving science.
Yeah. Anna, let's pick up on that topic, because sharing experience and sharing learnings, is there a role for industry to become involved in helping to continue to build the capabilities and the knowledge base in a regulatory agency, or for regulators worldwide? How can industry continue to play a role in supporting this educational evolution that's required in this new environment?
Yeah, I think it was echoed. I think earlier, just as Maxime said, and Marc and Tony, is basically earlier dialogues. So when, for example, from an industry perspective, when you get from your, like, for example, advanced therapies, the CAR-Ts. So when usually CAR-Ts are moving very fast, go from your phase 1 with your registrational study in phase 2. So at that point, you will need to, engage with your agencies, and also, to have this dialogue and go and talk about, your designs. but also to talk about how how is the scientific way of this this treatment works from a mode of action, but also from a modality perspective. So it's early dialogue that's got. Also, I think that one of the key things going to collaboration, because of course, targeting one agency at a time, is time consuming. One of the key things could be, industry. If we have a new program, we could have a round table, debrief. Like for example, the Access Consortium. We could, have an early dialogue for a new modality. I mean, for example, what we would like to see with the Access Consortium, because they have wider, spectrum in terms of therapeutic purpose. We would like them to see them, to have a pilot with conditional approval, which is not at the moment. So it's all about early dialogue, pilots, and kind of pushing the boundaries, and being flexible. I think collaboration, flexible, and basically pioneering. And I think for us, because I remember once when I was in the CAS forum, and we had one of the Danish quality assessors. They said, it's you, the companies, who have the science, you understand the science. So early dialogue, but also other fora, for example, the different scientific conferences, where we are bringing some of these, new, innovative ways of working. But I think more collaborative, meetings potentially, like Access, around the table, where we go and present our science, our early thinking. Yeah.
They're great suggestions, and I think somebody should take up your concept there as on the pilot. We have time for a question that's coming from the audience, and I'd like to pose this to Marc and to Maxime, and it has to do with oncology trials. What it says is, we've been asked, as oncology trials move towards earlier settings, and we're talking about things like neoadjuvant and adjuvant therapy trials. Can you talk a little bit about how regulators are preparing to look at surrogate endpoints? And this person suggests things like PCR, EFS, DFS, and other kinds of. So I think the question is, will there be a shift towards different kinds of endpoints as we're moving towards earlier kinds of settings? And Marc first, and then Maxime, the final word.
So I think we're going to be I think we're tasked already, but I think we're going to be tasked to be able to interpret early data, and I think it does get back to understanding the clinical condition and understanding if the endpoints that has been chosen is robust to be able to determine what you think the outcome is. And so it does speak to understanding the disease process and understanding where one can be flexible and are the numbers appropriate in terms of, for example, disease-free survival. Does that translate into overall survival in a particular condition? Does tumor growth change in terms of where you are? And so it may require that you have ad hoc advisors that provide you with these critical insights in terms of the clinical condition, because I don't think everybody can hold all this information in their head at once, despite the fact that we all are specialists in our own right. But I think if we don't move in this way, because the clinical trials are moving in this way, and so if we align the clinical trials to move in this way and have these endpoints, I think we need to get into collaboration with the industry in terms of who are putting these protocols together, in terms of making regulatory decisions up front and guiding what is a endpoint that we would be comfortable with that shows not only efficacy, but safety. And I think we may need to be a bit more robust in terms of the safety aspects, the downstream risks that may need a bit more work to be identified.
Well, it gives us lots of opportunities in the future. And Maxime, final thoughts?
I'm with Marc on that. I think as we move earlier in the treatment lines and in the treatment settings, I think the safety is key, because we're treating a majority of patients that for example, might actually be cured. So we're always looking to try to get earlier endpoints, but I think we have to remain cautious. We have built some confidence in earlier endpoints, and we are building on that. The conditional approvals was one way that we build that up. We allowed ourselves to sort of feel more confident in certain specific disease area and using surrogates. And we there's things like, for example, minimal residual disease that have made their way into clinical trials in the last 5 to 10 years that now feel a lot more comfortable. But when they started initially to be used, I think that's the kind of stuff that we were legitimately
skeptical about.
Yeah, interested, wanting to make sure that we were trusting something that was real. So, measuring drug activity versus clinical benefit is always a very tricky is a very tricky edge to work on.
Well, I want to thank everybody. As always, this has been a really fun and informative session. Just really great. Today we've covered a lot of ground, and typically what we like to do is leave you with a couple of recommendations, things that you can think about and maybe act on this afternoon or tomorrow that you can put into practice, whether you're a regulator, somebody from industry, an NGO. And here are a couple of takeaway actions that our presenters have suggested. One, obviously, is extrapolating early clinical data can potentially accelerate drug development. So think about that early clinical data. We just heard this wonderful discussion now about how it requires careful consideration. Companies planning to rely on earlier clinical data based on surrogates to obtain approval should consult early with regulators. We heard that over and over again. Have that conversation. And we should have robust, flexible pharmacovigilance. How do we follow these products up? How do we know that they are doing what they were intended to do? So I think in summary, these approaches can streamline the pathway to marketing for promising therapies, in particular for rare diseases and conditions with urgent unmet medical needs. So thank you very much again to our speakers. I want to just say that remember, a recording of this session will be archived shortly on the FR Path website, and we're sure that you can use this information in very practical ways. All the previous webinars are also archived on the FR Path website. Our next webinar will be Thursday, probably April 24th, when we will continue the conversation with a deeper dive into the topic that we started today around specialized regulatory pathways. We talked a little bit about conditional marketing authorizations, but there are emergency use pathways, all kinds of other pathways that we should be thinking about as alternatives to standard pathways. So we'll be doing a deep dive into that. Remember, free registration will be available at the FR Path website shortly and through our LinkedIn and email postings. Please keep an eye out for them. Once more, Mario and I would like to extend a very special thank you to our guests, to Tony, to Maxime, to Marc, and Anna. Thank you very much for taking your time out and sharing your thoughts and experiences with us. And Mario and I thank you for your interest and participation. All our best until we meet again.